Okay, good afternoon. Nice to see that so many people came. My name is Simon Anders, and this will be, as you probably all know, the lecture Applied Math in Biology, Single Cell Omics. If this is not the lecture you were expecting, then now is your time to run away. So you might have read the announcement, this HTML file that I made to explain the motivation behind the lecture. It's sort of to show to the mathematically oriented students from math, physics, scientific computing, and so on, that there's a lot of demand for scientists with mathematical background in the life sciences to give you a window into what's done in the life sciences. So I myself, I am trained as a physicist, also did my PhD still in theoretical physics, and then switched to bioinformatics or computational biology. And my aim here is to show you a bit what computational biology is about and why, in a way, it is more physics than biology. It is biology in the sense that it's a discipline which tries to learn about life and about how living organisms work. But it's also physics in the sense that if you consider physics as the science or the art of using mathematics to understand the world and to understand nature, then there are a lot of things are physics which you first don't think about it. And if you look at the areas where in science and in physicists work, you will notice that there's many areas where you simply need a mathematical understanding to work with data, and this is where physicists usually can work, and by extension also people with a similar background like mathematicians. And in a way, scientific computing is a major which is meant to do this. We are using mathematics to understand, to do science, and in my, of course, sort of biased view of being a physicist myself, this is physics. So, wow, we are nearly filling up this seminar room. Anybody else coming? Just close here. And of course, there's two ways how you can do this. I could give you a big overview of the open topics in biology at the moment, which is difficult for me because, of course, I don't have this overview either. But also when you just get a feeling of what the open question is, but not how you actually use math for this. So instead, I decided to pick one specific aspect of current biological research, not completely unsurprisingly the area where I personally work in, namely single-cell omics, because it's an area where many, which uses a lot of mathematical methods, very diverse mathematical methods. We will see linear algebra, we will see machine learning, we will use you'll see all the different areas of math that we pull in, some graph theory, some calculus, and we'll use all this, and you will quickly see that this is simply an area where you need a math background. So no disrespect to our biology colleagues, but if you do your undergrads in biology, you simply don't learn enough mathematical technique to really get deep into these things, which means that this is an area which is really filled by us more computational or more mathematical people. And hence, it's probably unsurprising that the majority of researchers working in these areas have a background in the more quantitative subjects. Unfortunately, today we won't do much mathematics, and you will have to wait at least till next week to see what all the math is about that I want to bring in, because obviously, first, assuming that you're all coming from another direction, I will have to give you a background on the biology we are doing. And maybe a quick question, who here is math students or a mathematician? Who's from scientific computing? Any physicists? Okay. Who else? What have we missed? Second? Biomath. Okay. We have a biomath major? Okay. Good. So you're the only one who actually comes from the other side and does and have a biology background, even though, of course, the biotechnology area has a bit more math than the biosciences. I see.
Yeah, and I'm also an exchange student, but study bioinformatics.
I see. And all of you in the master area, or any bachelor students around? Okay, also good. Doesn't really matter because from in the end, it is basically first year mathematics that we will mainly use. So first year bachelor mathematics. So if you're at the end of your bachelor, you're beginning at the master, doesn't really matter that much. So now I made a little course webpage. This is now the third time I give this course. Here is the webpage of last year, and I will sort of translate this to the new 2025 term and always move everything over there in the during the time. But if you want to sneak where we are getting at, you here see what I did last year. Of course, I will change a lot, but we'll see. So, and maybe the first thing we can look into is this biology primer, which maybe I first start telling it to you in the general thing. As we are looking, we will basically work with transcriptomics. So what this is about. You all know that all your cells in your body contain DNA, and they all contain the same copy of your complete genetic information in form of long DNA molecules called chromosomes. And so in that sense, the total information in all the cells of your body are one and the same in all the cells. But nevertheless, you have very different cells. You have skin cells, you have muscle cells, you have neurons, you have blood, various kinds of blood cells, etc. And all these cells do something different, even though they all have the same program, if you consider the DNA as a program of a cell. And the reason is obviously that, to now use computer science terminology, that different parts of the DNA is executed of a program, and depending on what state the cell is, it has different parts of the DNA active and other parts are inactive. And so in that sense, the difference between cells which all have the same genetic information lies in which genes, which parts of the DNA are active in the sense that the cell follows the instruction it finds there. And the most important way how a cell uses the DNA is the so-called transcription. The cell makes short copies of the interesting pieces of DNA and moves them outside the cell nucleus to the cell plasma, so the whole liquid that fills the cell. And there this transcript, which is a copy of a short piece of DNA, then serves as instruction for the machinery in the cell to do something. And the most important kind of instructions are protein coding RNA, which contains blueprints, instructions how to assemble proteins. And proteins, again, are, as you might probably all know, sort of little complicatedly shaped big molecules which either make the structural backbone of cells and bodies, or which are little molecular machines, which are the tools and machines which do all the chemical work in the cell. And the DNA tells the cell how to assemble them. And depending on what state the cell is in, the cell produces different proteins and uses different proteins. And if you now have two cells and you want to understand what's the difference between these two cells, an easiest way is the ideal way to really understand what's going on in the cell would be to know which proteins are currently in the cell and what are they doing. But this is technically very complicated to identify the protein composition of the cells, to identify all the other things. But what is much more easy is to sequence the RNA, to sequence all these transcripts and see which instructions is the cell currently following. And that already gives you a good idea, with a good proxy to understand the state of a cell. So if, for example, you do medical research and you want to understand what might be the molecular underpinning of a certain disease, you might collect tissue samples from patients who have a disease and from healthy control people. And when you simply compare these two samples with respect to which parts of a DNA is active, which instructions are currently being followed, in order to find out which instruction might be too active or which might be not active enough, and so get a clue about what actually went wrong in order to cause the disease. And therefore, this is maybe the easiest way currently in modern molecular biology to get a lot of information about a sample, a tissue sample or sample of cells. And because we get so many informations about this, and all this information is sort of in a homogeneous fashion, as we will see in a moment, this is something where we can easily work with computational techniques. And we will see in the next few weeks how this kind of information looks like and why it's so amenable to be interpreted using methods from linear algebra. Now, to start off, I made here a couple of fact sheets, many of which you probably still remember from high school biology, but let's quickly go through them to make sure that we are all on the same page. So we start off with DNA, which is the type, stands for desoxyribonucleic acid, which is the famous double helix structure, which contains all the information in your cells. And DNA is a polymer, so that means that it's made up of a long chain of repeating elements, which we call the nucleotides. And just to DNA, let's quickly look at a picture. I should probably have found one beforehand, but you've all seen these pictures often enough. Here's a nice one where we can see how this is. This is the actual chemical composition of a DNA, and this is sort of a simplified idea. You have your backbone, which is one helix and another helix, and this backbone connects all the different nucleotides, and the nucleotides sit in the middle, and they only find space to sit in the middle if the two nucleotides, which come from either side, fit in a certain way, that where the nucleotide adenine is always paired with thymine and guanine always with cytosine. And this is the famous base pairing, which means that each of the two helices has the whole information, and we can easily make a copy by tearing the double helix into two single strands, and then building a new strand by putting in the missing bases. Because what you can also obviously see is that each base here contains an information of 2 bits, because we have a total of 4 possibilities. So this is what we start off. We have the 4 nucleotides, which have these names A, C, G, and T, adenine, cytosine, guanine, and thymine, which are always paired, A with T, C with G. And this means that whenever you have a DNA molecule, you can either read along one of the 2 halves of a double helix, or you can read along the other half. And because they are mirror symmetric, one is curling upside, the other is curling downside, as it's shown here by these arrows. So chemically, the bonds here which connect one with the next are asymmetric, so there's a direction. And this means this is one direction how you can read the DNA following this line, or this is the other direction following the other line. The chemists always denote the end where you start with 5' and the end where you end with 3'. And hence every DNA sequence has 2 equivalent ways how to put it, either forward, A, G, G, C, T, or the backward one, where we would say this is the sequence A, C, T, G, A. And you have this operation called the reverse complementing, which swaps A's for T's and G's for C's. Oh, there's a typo here. And also swaps the direction. And DNA duplication means the DNA is broken up into its 2 halves, and the special protein, the polymerase, comes and builds the reverse complement. Yeah?
Is there an intuitive reason why one starts from the bottom and goes to the top and the other one does the reverse?
I guess it's one of the, good question. I would say it should, if you stand long enough in front of a 3D model, you sort of notice how this works. So you have here the sugar phosphate backbone, which, let's see if we, here in a backbone, if we have a picture of this thing, now we get the overview. So here you see how this works. You have here the sugar and here the phosphate, and it's some kind of asymmetric thing. It just turns out that you need to not make a mirror, but a point symmetric operation. So make a, how do you call this in English? A Punktspiegelung in German. But you know what I mean. In order to go from one strand to the other, and of course replace the A's with the C's. I guess this simply evolved in the sense. So you ask for an intuitive reason, and in biology, there's an old saying in biology, nothing in biology makes any sense except for in the light of evolution, which is again the case here. If it weren't like that, it wouldn't be mechanistically easy to make copies, and therefore it has to be like this, otherwise it wouldn't have been fixed and life wouldn't have started with this construction. So, but of course, this is all speculation because that's so long ago we don't know how it started. We only know that these are mechanisms which are common to all living things in the world. And to make these copies, I already said, there's a specific enzyme called the polymerase, which separates the strands and puts it together. Something I wanted to show you, polymerase animation, is a little video because sometimes it helps to get an understanding of how, to get a bit of an idea what we mean by enzymes. It's useful to have a bit of view of what's happening here. Here you see our DNA strand, which now for, to simplify the animation, they depict it now untwisted, which is of course not what happens in reality. And now they show us here how the DNA is here broken up into two halves by this helicase, which is one specific enzyme which does something. And it's called a helicase because also it has to undo the twist of a helix. Then you see here how now another enzyme comes, which is called a primase. You see they all end on A's, which makes a short part double-stranded because the next enzyme which now comes, the polymerase, can only work if there's already a little piece of double-stranded DNA and then goes from double to single strand. And now you see how there is just floating around in solution the individual nucleotides, which just float around and diffuse and might accidentally hit here so that they fit. And if they fit, then the polymerase fixates this and moves a bit further. In this way, builds the longer strand. So this is going on whenever a cell in the body divides into two halves. And obviously, because you split it into two and you want to make two copies out of one, you have these two copies. And because the polymerase can only go in one direction, namely in the arrow direction, as we've just said, in one way it's backwards and one is forward. In the so-called leading strand here works easy. It just does what you see, always putting in something new. Here the lagging strand has the problem that it always has to add a little thing and then go forwards to what is already done. Here, let's make this when the polymerase comes, adds this, and so on. So this is what we mean by molecular engines. We make really complicated molecular constructions, and this becomes even more impressive if you look at these nice, what's their name? Animations that WEHI, that's in the Weizmann, that's an institute in Melbourne in Australia, and they have this one working group which is famous to make these animations which are actually produced by actual molecular simulations of the individual constituents of all this machinery in the in solution with all the water molecules filling around. And here you can really see what it means for an enzyme to work. You can see that these enzymes are machines shaped in a complicated way that can change their conformation, can jump around, bind with each other, interact, and do complicated conformal changes. But each of these colored things is one protein, and the crucial idea of a protein is, even though it seems to have a complicated structure, it's actually a one-dimensional chain. The same way how DNA is a one-dimensional chain, proteins are also a one-dimensional chain. See how we get here a picture, where you see that whenever, where you can see that you have these individual components called the amino acids, which are here the purple things, which are connected to each other with these always so-called peptide bonds, which is always a carbon, nitrogen, another carbon, and here an oxygen. And in that way, you have 20, how many are they? 20 something different different amino acids which can be chained together in one long chain of maybe hundreds or even thousands of amino acids, and a protein is defined by the order in which you chain them together. So let me show you first here the famous genetic code. These 20 chemical molecules around are the 20 amino acids that are used in living cells to produce proteins by chaining them together in a long chain of 30 of hundreds or even thousands of them. And then once this chain has been built, something magic happens, because some of these things are hydrophobic, some are hydrophilic, some are polar, some are unpolar, some are electrically charged, others are neutral, and so they attract each other or repulse each other, or they attract the water dipole of the water molecules and hence pulled outside, or they are rejected by the water and hence are pushed inside this whole conglomerate. And in this way, the chain coils up into a complicated shape, which we call the tertiary structure of the protein. And so indirectly, the sequence encodes also a shape. And this shape is what makes the protein work, because it gives the protein the right shape to get levers and tools and all kinds of ways to, as you've just seen video before, wrap around the thing it has to work on, or move along it, or push it, or do whatever. Yeah.
But it sounds like the shape should be unique. Why would just these hydrophobic, hydrophilic, etc. properties be unique?
This is a bit an interesting question. A priori, you wouldn't assume, but a protein only functions if it's unique. So evolution has constructed proteins such they can only fold in a unique way. Imagine in a way you have some origami thing with lots of little magnets in your origami, and you try to make sure that the magnets always snap in one specific way, and every time you try it, they snap a little bit differently, and you get a different shape, and eventually find out a way which reliably always folds into the same shape. And then you have a well-working protein. And this seems to have been the big work of evolution in the first 1 billion years of life on Earth, to figure out suitable protein sequences which do that. So now I'm jumping ahead a bit here. Let's go on. Here's but maybe it's good that I jump ahead, because in biology they have something they call the central dogma. The central dogma is a bit a bad term, because the guy who came up with the word later admitted that he didn't know what the word dogma meant. What he actually means is the central idea of how biology works. You have your DNA. This DNA contains short pieces which have the construction plan for one protein, which we call a gene. This is copied into another kind of molecule called RNA, which is chemically very similar to RNA, only the sugar has 2 extra oxygens. That's why one is called desoxyribonucleic acid and the other ribonucleic acid. And it doesn't nicely fold into double strand anymore, but typically stays single-stranded. So the cell makes a copy of a stretch of DNA, copying it into this other biological form, the RNA. This then is moved into this then goes out into the whole volume of a cell, where another kind of molecular machine, this ribosome, finds it, and the ribosome follows the explanation in this thing to build the protein. And the way how the ribosome does it is by using the so-called genetic code, which you can see here. You see each DNA sequence, when coding for a protein, consists of triplets, so of triplets of 3 bases, and each triplet codes for one specific amino acid. Here, for example, if you see a G followed by an A followed by a G, when this means next is glutamate acid. If when the next 3 bases is GCA, when the ribosome knows I should pick out a glutamate acid somewhere and chain to it an alanine. And then I should chain to it whatever the next triplet says. And in this way, the protein, the ribosome always reads 3 DNA, 3 letters from the sequence of this thing, finds the corresponding amino acid, and chains it to the part of the chain it has already built. There's again a wonderful Vehi translation. So this is not so much important for the mathematics, but A, they are really nice, and B, it helps you to see a bit how this works. So here we have, this here is the nucleus, the inner part of a cell, and this is an RNA molecule. So one of these single-stranded things which just came out, and here we have the lower half of a ribosome, and now the upper half of a ribosome, which just folded around it. And these yellow things, they are the individual amino acids, always coupled with the so-called tRNA, which is a little marker of which triplet they belong to. And they float around, and the ribosome, as you can see, always moves ahead, always 3 bases along this chain, and then waits for the correct correct amino acid to diffuse by, and if it gets the right one diffusing by, it grabs it and attaches it to the chain. And in the end, here the red thing is the chain of amino acids which comes out. And here the green thing, the big green thing is the so-called transfer RNA, which is the marker, and somewhere next to it is then the red thing. Again, you can see this amazing complicated molecular mechanism, how there's really some kind of levers going on which grab something, shift it around, and do complicated mechanical things in microscopic little things. What you can also here see, the red one is the actual amino acid, the green thing is the holder for the amino acid, so that the RNA, the ribosome can grab it and put it in. And in the end, you get now the protein coming out here as a chain. And while it comes out here, it already starts folding together and getting into its shape. And obviously, it only folds correctly if it comes out in the correct speed and correct order and so on. And with this, we now have what we call the central dogma, that the way how biological material is produced works the way that first a gene is active in some cell, which means that a molecule called polymerase comes and makes a copy of this gene, copying DNA into RNA. When this RNA is moved somewhere where the ribosome can find it, and when the ribosome reads off instruction on the RNA and produces a protein. And among the proteins which you can produce that way are also all the machines which you need to make the polymerase and so on.
Mentioning how the RNA is a copy of the DNA and we read in triplets of bases, it seems to me the bases differ from RNA and DNA.
The thing which I've mapped over is while copying the DNA into RNA, A, C's, and G's stay as they are, and T's are changed into a new molecule called uracil. This is why you always write EU instead of a T, but this is one of these minor details. So let's quickly go now here through my notes, what we now, to put this now into another context so that you see what we need to know. As I said, all the cell's information is in the DNA. We have talked about base pairing and so on. We have talked about these enzymes called polymerases, which can break open a DNA, the helix, and make a copy of one. And the copy can either be again DNA, that's the thing you need if a cell divides, and you need 2 copies of its material for each of the 2 daughter cells, or it can make a copy into RNA, that's when only a short stretch, which is then later handed over to a ribosome. And and in eukaryotes, which is all more complicated things, everything which is not a simple bacterium, you have a nucleus in the middle of a cell, which contains the DNA, and it is organized in long molecules which are called chromosomes and which are linear. And in bacteria, it's simply a small little loop. And there's also, you might remember from high school, the mitochondria, which is sort of cells which live in eukaryotic bacteria which lives in cells, and they also contain a loop. And because we will work with them, we need to know how they are used in information informatically, and here we simply number the chromosome from the longest to the shortest by numbers from 1 to 22, in the case of humans who has 23 chromosomes. Otherwise, some species have more, some less, and then of course there's the sex chromosomes, which don't get a number but an X or a Y. And everybody has slightly different DNA, which is what makes us genetically distinct, but the differences are not so big. Maybe only a 0.1% or even less. Maybe every 30th or 40th base is different from one person to the others. The main part is the same. So it makes sense to write down the typical sequence of chromosome number 1 of a human, which is then simply a text file with 100 million letters, which are either A, C, G, and T. And you can download exactly like this as one long text file just with A, C, Gs, and Ts. And of course, this text file will not correspond to one specific, or maybe it corresponds to one single specific individual, the one who was used to make this thing. In practice, it's more an average mixture, a consensus through different ones, and then you get other databases where you can look up for each base in these human genomes, in the 3 billion bases that the human genome has. For each of these 3 billion bases, you can look up, is this one of those bases which are really exactly the same in nearly all humans, or is this one of those which are maybe an A in 80% and a C in 20% of the people? And all this is 23andMe stuff, which looks at heritage and so on. They use these kind of things. So I talked about RNA, which are copies of small stretches of DNA, which can then be moved out of a nucleus in order to instruct ribosomes. And a stretch which can be copied is called a gene. And obviously it has to have a beginning, a marker which tells the polymerase this is a place where a gene begins. So if you want to transcribe, start exactly here. That's called the promoter. And at the end, there's another marker which marks the end of a gene. And this making a copy is something we call transcription. And the produced RNA is called a transcript or a messenger RNA. Messenger RNA because it's a message from the nucleus to the cell plasma. And yes, the word gene had changed its meaning a number of times, and specifically have to distinguish from the meaning of the word gene in the sense of Mendel, that you might remember from high school, where you talk about dominant and recessive stuff, and the meaning of a gene in the sense of a piece of a DNA that can get transcribed into one RNA molecules. These two meanings are surprisingly similar, but not quite similar. But to explain why they are not similar, I would have to add a lot of more background. So for now, we simply say most of these genes, actually not most, but the most important ones, are blueprints for proteins, and we call them the protein coding genes. The human genome, as I've just said, has 22 chromosomes, 23 chromosomes, with a total of 3 billion base pairs, and it contains around 23,000 protein coding genes. There's another 60,000 non-coding genes which seem to get transcribed, but do not contain instructions to make a protein. And for a long time, people put them aside and said, we don't understand them, let's simply ignore them. Recently, people have a feeling that maybe these might be important. I talked to, of course, as we are now also computer scientists here, we have to think about conventions. So if we have such a chromosome, as I've just told you, each chromosome can be read in two directions, either following one helix or following the other helix, and one is the reverse complement from the other. So if you want to put this into a text file, which end do you start? And where this is solved by a simple convention, these chromosomes have a little part in the middle called the centromere. You might remember these pictures from high school where the chromosomes are X-shaped and where they stick together at the centromere, and the arms are not the same length. So we simply say the shorter arm, the petite arm or P arm, comes to the left, and we read the sequence from the shorter to the longer arm. And if whenever we talk about RNA, we read the RNA in exactly the sequence, the order in which they are transcribed with polymerase and the order in which they are read by the ribosome, because these two directions are the same. And they don't have to be the same direction as the DNA polymerase copies this thing. And for reasons that only the biochemists can explain to you, the starting end is called the 5' end, and the ending end is called the 3' end. So proteins, I told you, they are polymers formed as a chain of amino acids. And there's proteins which catalyze chemical reactions, and they are called enzymes. And we've just seen a lot of these chemical reactions happening in these little animations whenever the DNA is opened, copied, and so on. These are all biochemical reactions. Hence, they are all catalyzed by enzymes, and all these little things that you've seen are enzymes. But there's also a lot of other things which do scaffolding, which form the membrane of a cell, which form the cytoskeleton, which keeps it propped up and from falling apart. And all this scaffolding, they are called structural proteins. And as I told you, there's 20 proteinogenic amino acids. Two special ones, which we skipped over. These are these 20 ones, and each code codes for one of them. But as you can imagine, we have 4 bases and triplets of bases. 4 to the 3 is 64. So it's 64 different codes, but only 21 meanings. So the code is redundant. There's usually several codes which code for the same amino acids. There's 2 codes which are special. This code here, which is not an amino acid, but it's called the stop code, which tells the ribosome that it's finished and that it should just clip off a chain and eject it. And the start codon, which tells the ribosome start here with it. The start codon is actually this one here, which also codes for methionine, which explains why all proteins start with this amino acid, unless some other enzyme has clipped it off later. So, and what else do we need here? And exactly, and hence the list of amino acids which have to be chained together by the ribosome, they define the protein. And if evolution has made the protein well, which is usually the case, the protein will always fold in one and the same shape. Sometimes it doesn't, because maybe it was too hot and the vibration from the heat caused it to misfold, or you have a prion, or you have something else, and this can be cause for disease. And the assembly is done by this special molecular machine, the ribosome, which we've just seen in process. And we call this the translation because it translates according to the code. And yes, and in the end they fold. And the reason why they fold is because each amino acid is a bit different. The genetic code I've also shown you here. And I've talked to you about the types of genes and transcripts which we find. We will mostly talk about the protein coding genes, which always have 3 parts. In the middle is the coding sequence, which contains the amino acids, but there's a bit of stuff before and afterwards, which are not seen by the ribosome, but which are seen by other proteins, which might see this as marker to grab the RNA and move it somewhere in the cell, or degrade it, or destroy it, or freeze it, or whatsoever. And then there's also many other transcripts, ribosomal genes, which make up part of the RNA, transfer RNA, which are these little adapters which hold the amino acids, and many other things. You might have heard about splicing in the human, especially in more complicated animals. Typically, a protein coding gene looks a bit strange in the DNA. It always has short stretches which contain transcript, and then comes long stretches which have to be skipped, and then short stretches again. And so the polymerase makes this copy of this long thing, and then it removes these things which shouldn't be part of it, which should be skipped, by bringing the whole chain together there and clipping out the loop of this extra thing. That's called splicing, and in this way, you get from the RNA to the much shorter mRNA. You don't have to remember this now, but we will get back onto this later. But it also means that you can, from the same gene, you can assemble different transcripts by either including some parts or removing some parts. Now, the big part which is missing here is how does a cell actually know which genes to transcribe? I told you in the beginning that some cells, different cells do very different things. And for different functionalities, you need different proteins. For example, if you have a skin cell or a hair cell, you need a lot of keratin, which is this hard substance which makes up the outer layer of your skin or your hairs. Or if you're an osteoclast, that is a bone cell, you need to have the proteins which make bone. Or if you're a red blood cell, you need hemoglobin to transport oxygen. So how does the cell know that it has to transcribe the hemoglobin gene because it's a blood cell, but not the keratin gene because it isn't a hair cell, or vice versa? This is called gene regulation. We want to understand how genes, we want to understand how a cell knows which genes should be activated. In a way, this is the big question in the whole thing, and we know a lot of little parts, but we are completely missing the full picture. What we know is this. The polymerase doesn't simply go through the DNA and says, oh, here's a gene, let's transcribe it. It needs to be recruited. So other proteins need to already sit there at the beginning of a gene and wave to the polymerase, and as the biologists say, recruit it and set it to work there. And so there is things called transcription factors, which are proteins which go through the DNA and they look for the motif. They look for a specific sequence in the DNA, and when they find this, they bind there. And then once they are bound there, they may be able to activate these genes. And there's seems to be some complicated combinatorial code that maybe if a transcription, that a certain gene might have motifs for the transcription factor X, Y, and Z, and whenever the transcription factor X and Z is present, and both of them have found their motif and bind there and sit where they can together pull in the polymerase, but not one of them. And if Y sits at its motif, then it sort of blocks the other two and keeps it. So you get some kind of Boolean logic. This gene gets transcribed if the transcription factor X and Z and not Y is present in the nucleus. And now, of course, these transcription factors are also proteins, so you can see where this is going. We have a complicated feedback loop. Some protein might code for a protein, some gene might code for a protein, which is a transcription factor, which switches another gene on, which switches a third gene on, which again regulates the first gene. And so you have these loops, and you might get positive feedback loops where genes stabilize it. Gene A activates gene B, and gene B activates gene A. So once one of them is activated, we keep each other alive. And you might have negative feedback loops where when a gene notices that there's too much of it, it switches itself off, maybe via a detour of activating something else, which switches the first thing off. Another important part here is the so-called post-translational modification. After the ribosome has built the protein, it doesn't stay as it is. There's other enzymes which can come, find a protein, and change something about the protein. There might be the most important class of these things is called kinases. A kinase is a protein which recognizes a specific class of other proteins, maybe because they have a certain shape. They might have somewhere in their complicated folded shape some kind of hook, and the kinase, whenever it sees a protein with this hook, it might bind to this hook by clamping to it, and then it reaches out with some other parts of its molecular machinery and adds a phosphate group, so a phosphor atom with 3 oxygen atoms at some specific place. And when you add a phosphate group to a protein, this often changes something. It might change the biochemical properties of the amino acid where you added it, maybe adds a charge or removes a charge or changes the charge, so that the whole folding changes. So the kinase, by adding this phosphate, wrong way around. A phosphatase adds a phosphate, a kinase removes a phosphate. But a lot of stuff happens in biology by one enzyme adding or removing phosphates or other stuff from other enzymes. And whenever this happens, the whole folding changes and the molecule suddenly snaps into a new shape. And this can activate and deactivate them. So there's many proteins which float around and first do nothing. And when some kinase or phosphatase comes along, and activates this enzyme. And now the enzyme is active and can, for example, do its role as transcription factor. So you have another level of regularity logic where proteins activate and deactivate other proteins. And you have whole cascades of protein A activates protein B, which activates protein C, which blocks protein D, and so hinders it from activating protein E, etc. Often these cascades start at the cell membrane, where the cell somehow recognizing some messenger coming from outside, maybe a hormone or a message that another cell sends it in order to tell the cell to do something. And all these things we can't see if we do transcriptomics, but we see the consequences. We don't see that a transcription factor was activated, but we see that its target genes get now transcribed. And in that sense, we usually assume that the state of a cell would be best described by saying which proteins are in the cells, which of them are in their active conformation and which are in their inactive conformation, or often they have lots of different conformations and can be switched from one to the other by others, and we would want to describe the state as this, but what we get instead is the complement of RNA in the cell, because that's much easier to measure, as we will see in a moment. It's really easy to take a cell and find out which RNA is in the cell, and then make a count. We see 20 copies of this gene, 3 copies of that gene, etc. Yeah.
I was understanding previously that the protein folding was unique. Now to my understanding, these additions of phosphate groups changes the
Yes,
folding.
this is called allostery, and allosteric change is unique in the sense that the raw protein with unmodified things folds in one thing, but once you add some modification groups to it, folding changes. And that's a very important mechanism in biochemistry. So, now the way how I describe it here is maybe not the way how it's usually described in biology. I more try to describe it from the computer scientists' perspective, because I hope that you get a feeling or an image now in your mind about how a cell is a complicated computing system, or for the computer scientists in here, a state machine, or a network of many different state machines which all interact and change each other and have complicated feed-forward and feedback loops. And I don't know who of you has ever done any kind of systems theory or dynamic theory. Anybody done anything like this, control theory, systems, dynamics or so? Pretty, but maybe you can imagine this kind of simulations that you do more in physics. If you really wanted to simulate all these things, you imagine a complicated ODE system where where you have ordinary differential equations which describe all these biochemical reactions, where depending on the concentration of all the various different proteins, the concentration of other proteins change. So people then write down ODE systems where you say, we have one variable which tells us the concentration of the unmodified protein and of a modified protein, and a third variable which tells us the concentration of a modifying enzyme, and then you can, and maybe a fourth variable which tells us for the enzyme which undoes the modification, and then you can imagine that you can write down a differential equation which shows you how the concentration of unmodified and modified protein changes by them interconverting into each other, depending on the concentration of the modifying and of the enzymes which do the modification. If the activity of these again depend on the original enzyme, then you can see how you get a complicated ODE system with complicated multi-stable behavior. But who has worked with ODE systems? Okay, then you can probably roughly imagine how you think. A cell is full of strange attractors. So, what do we now, as the next step, I thought, the next thing I wanted to show here is a quick look at why is it so easy? I told you it's much harder to find out which proteins are in a cell than to find out which DNA and RNA is in a cell. And to understand this, I give you a very quick overview of the technological basis of this, sequencing. When you have a DNA molecule, you can read off its sequence. I give you a long molecule, and you have a machine which says this molecule is a DNA helix with the order A, C, G, G, A, C, C, T, and so on. The crucial invention to do this is Sanger sequencing, which was invented by Fred Sanger in 1977, and which allows us to read off a molecule. And the problem was you need to purify this molecule, so make millions of copies of the same molecule, and then you can maybe read a few hundred bases of this, and then you have to start over. So it's a long, slow, laborious process. And at the end of the 20th century, the famous Human Genome Project was working on using this technique to slowly read off all the 3 billion bases of the human genome. Around that time, as I said, the other crucial thing is the polymerase chain reaction, PCR, which is a mechanism which allows you to make billions of copies of one DNA molecule, simply by using the fact that the polymerase, so this molecule which works in cells to duplicate the DNA in order to prepare for a division of a cell into 2 daughter cells, this thing also works in a test tube. And so you can put it in, and it will take every molecule it finds and make 2 copies, and makes 2 copies of these, and you get an exponential blow up. And that's what makes it so easy to get large amounts of one sequence, which you can run through with Sanger sequencers. But the catch of them is they work in solution with liquids. So you have to make sure that there's only 1 copies of one and the same molecule in it. Otherwise, you read of a mixture of several of them, and they are all blurred. And you always read the first molecule is ACG or T, and the second is also ACG or T, and so on. And then in the early 2000s, this idea came up that you can fix the DNA fragment on the surface of a glycerol of some kind of functionalized chip. So you have some so-called flow cell, which is just some block, and I don't go into technique here, but you come in with a mixture of many different DNA pieces. Maybe you took the DNA from some cell or from some organism, manched it up and split it into millions of little pieces, and you spread it out, and maybe one piece stops here, and one piece ends up here, and one here, and so on. And the surface of this chip is somehow chemically treated so that these things stick to it. And then by a trick, which I now don't really want to get into details, but it's a really beautiful idea, but it requires me to explain PCR first, so let's not do it now. You can do again this polymerase chain reaction, where the polymerase comes and makes copies, and the copies attach right next to it. And then after you do this 10 times, each of these things will have turned into 1,000 or 1,024 things. And now you have lots of spots, and each spot contains a few thousand copies of the same molecule, and they are all well separated from each other if you spread them out well beforehand. And now the trick in the Sanger sequencing, and again, I don't have the time now to explain it, but if you're interested, you find plenty of YouTube videos which show you this with a beautiful animation. With the Sanger sequencing, you always let the polymerase add one nucleotide and then stop, and add the next nucleotide and then stop. And each nucleotide is connected with a fluorescent marker, so that these 1,000 things, which all get the same nucleotide attached, might all light up red because an A was added, and these might light up green as a C is added. So you watch under a microscope how all these things how one nucleotide after the other gets added, and each one lights up in a color according to its letter, and you just have to record the sequence of colors that you see from each of these spots, and in that way you can sequence all these things simultaneously. Which in the upshot means this whole thing was, of course, now automatized with machines, and the modern sequencing machines, they can sequence typically around 100 million such pieces to maybe 100 bases within a day or two. So you split up your molecule, your DNA into millions of pieces, which you spread out over this sequence. And when the machine runs for maybe 2 days, and after these 2 days, it has read off from each of these pieces the first 200 base pairs. And so biologists always talk about base pairs, because when you read off one base, you of course also know the partner base on the other strand. And what this means, if you want to figure out now the human genome, or you have some new species that you've just discovered, and you want to know its whole genome, all its 3 billion base pairs, what you can do is split this up into millions of fragments, each maybe a few hundred base pairs long, put it into such a machine, and then you get a text file with 100 million lines, each line containing around 100 bases. So a string of length 100 of ACGs and Ts. And now you have to puzzle them together in the right order by looking for overlapping things, and along these overlaps puzzle them. You can quickly imagine this sort of scales quadratically. So at the time when this idea came up in the 1990s, everybody said, yeah, this won't work, a computer will run years on puzzling this together. But Moore's law helped. And around 2002, people suddenly realized actually computers are fast enough to do this assembly in a couple of weeks. And this then had this funny effect that in the, and ever since then, people are arbitrarily fast. You might have heard of Craig Venter, who got rich by having been the first guy to realize that computers are now fast enough to do this assembly by puzzling together these pieces. But that's now over 20 years ago. So, this is what I've written here. And this is called high throughput sequencing, or some people call it next generation sequencing, because it was the next generation after this. I don't like the term next generation sequencing because this is now 25 years ago. So maybe we should finally call it present generation sequencing, but you still will find the term NGS quite often. So, and the first thing you can do is you have a new organism and you want to know which DNA, what DNA is in this organism, what is the genome of this organism, and you want to make what we call a reference assembly. And the next thing is you might have as an individuum from a sequence species, maybe a human being, maybe you want to know how your sequence, where your genome differs from the reference assembly. Then, of course, you can take some arbitrary cell from your body, extract the DNA, sequence it, and this time you don't have to puzzle it together. You just take each 100 base pair read. So we call this a read, such a reading of the sequence from one of these clusters. You take the read, you go through this 4 billion base pair long, so 3 billion base pair long text file with all the bases of a reference, and find the place where your piece that you just had matches or nearly matches. And once you found that, you can say, okay, it only matches at one place, and this place it matches not perfectly, but these two bases are different. So this is the two places where I differ, where my genome differs from the reference genome at this site. And you do this for all these pieces, and then in the end you get a long list of places where you difference from the reference genome. And if you're unlucky, you might find a specific places, for example, the place which is marked BRCA. You will find a specific mutation which tells you that you have the infamous BRCA mutation, which gives you, if you happen to be a woman, a 50% risk of actually getting breast cancer. But there's only very few things which are that clear of a disease risk. So, but the other thing which we can do instead of sequencing DNA, we sequence the RNA. We take our cells, and we take out all the RNA, so all these transcripts, and we sequence them. Technically, we first have to reverse the transcribe the RNA back into DNA because the machine can't deal with RNA, but that's an easy trick in the biologist's lab box of tricks of the lab. Now, doing this, you get, you now will find something new. Before, if we took two different samples from different parts of the same animal, you will get the same sequence. If I sequence part of my blood and sequence part of my skin, I will get the same DNA. But as I said, we will get very different RNA. Also, this time we won't be interested in actually looking at the actual sequence. If I get a read, all I want to know is which gene was how active. So I take all the reads that I read and try to find where they come in the genome and which gene is there. Because you can imagine, maybe I show you at the moment quickly how this works. So this is what one of the workhorses, the Ensemble Genome Browser, where I can now go here. Here we have this place, and look. Let's take this one here. No, this thing is again too small. Here we see a stretch of chromosome 13 from position 32 million to position 32.4 million. And along this stretch, we see here the part which is this BRCA gene, which codes for one enzyme that the body needs in order to prevent cancer. And here, these are the exons, so the parts which are actually protein coding, which contain amino acid sequence, and these are the introns which need to be skipped. Now, if I get a read, and I look for the read, and I find the read matches to this part of a DNA sequence. Let's zoom in a little bit in this small thing here. So here now I've zoomed in, and here at the bottom I should probably see the actual sequence at this part, if I've zoomed in deep enough. And when I see this, then I might say, okay, this read is from the BRCA gene. And in the end, I make a big checklist where for each gene of these 50,000 genes in the human genome, I have a number which says, in my sample, I have seen 325 reads from this gene, 2,000 reads from that gene, etc., and so on. And in that way, you can then you get what we call a count vector. So, what else do I have here? Yeah.
In the discussion of sequencing RNA, it sounds to me like the thing we're interested in are these strands of RNA that ribosomes receive to assemble into.
Yeah.
But you mentioned other types of RNA, this tRNA and everything. Does this method effectively exclude?
Not voluntarily. We need to do a few tricks. The tRNA, the small RNA, we get out of her very easily because there's some trick which is called size selection, that we only get pieces which have the right size, between 100 and 300 base pair. The bigger issue is all the parts which make the ribosomes. We also have to get rid of them. One easy trick is, that we will get into much more detail later, is when the RNA translates normal messenger RNA, in all but a few exceptions, once it's finished, it adds a couple of extra A's, the so-called polyadenine tail. And we can grab only these. But I come to this maybe a bit later. Now, what I will now show you is just that you get a quick feeling. No, I have to talk about single cell RNA. Is about bulk and single cell RNAs yet. So here you can read this a bit more. So our step is we take each read, find it in the genome, and then we get any information which gene this is from. And in the end, we get a count which tells us for each gene how often we have to see a gene, a read that seems to have come from the part of a genome where this gene sits. And usually, traditionally, you do this to compare different samples. Maybe I want to find something out about a skin disease. Let's say you're a researcher interested in psoriasis, and you have skin samples from 10 patients with psoriasis, and you always have have from each patient, you have one sample from the afflicted parts of the skin and one sample from a healthy part of the skin. And now you want to find out which genes are more active in the afflicted parts than in the healthy parts. So what you will now do is that for each gene, you calculate the average ratio between the count for this gene in the afflicted versus the count for the healthy rate. Average these ratios, these rates over all your different patients that you have, and then you sort it to find those genes where the expression, so the abundance, is much higher in the afflicted parts than in the healthy parts, and say, yes, these are probably genes which are connected to the cause of a disease. Or maybe you might want to compare healthy, non-afflicted skin samples from psoriasis patients with non-afflicted skin samples from totally healthy control subjects in order to find out what genes might always be off in patients compared to control subjects. And this is the standard mode how you work with this kind of data. Usually the first step is always compare the counts between treated and control groups, or between samples from people with a disease versus healthy people, to find something. But if you do this, we have one fundamental problem. If I now find that a certain gene is strongly expressed, say, in the afflicted samples and not in the healthy skin samples, we still don't know whether this means that all the cells in this afflicted sample expressed the gene at a higher level, or only some of them. And this is important because a typical skin sample, you might know how doctors do this, they just cut out a whole piece of skin with a puncher. And this whole piece, which contains a mixture of many different cell types, actual skin cells, so actually the cells which make the skin surface, hair follicle cells, which makes the little hairs on your thing, muscle cells, which make your hair stand up in case you have cold, nerve cells, which connect to all your receptor cells to make you sense touch and heat and cold and pain, endothelial cells, which line the blood vessels, which bring the blood supply to the skin. So there's dozens of different types of cells. In which of these cell types is now the one which has this higher gene? Because after all, we just take all these cells, we break open the cells by digesting or lysing their cell membrane, and when we mix the RNA from all the cells, we won't know which RNA molecule was from which cell afterwards. If we could keep this information, we would be in a much better state. And we can do that. And this is sort of a new idea which came up in the last 10 years, 5 to 10 years, this technology really matured, that you don't take a whole sample of millions of cells, you just take 1,000 cells. But for each of these 1,000 cells, you keep a tracker. Each cell gets some kind of number, some random number, or actually a random DNA barcode. First cell might be the cell ACGGAT, the next cell gets the random sequence TTACG, and this random sequence is attached to each RNA molecule, so that when we sequence it afterwards, we not only know which gene the cell is from, the read is from, but also which cell it is from. And with that, we can now do a lot of more interesting analyses. But we also run into a lot of interesting mathematical challenges. And before going here through the back of this, I show you now, make a quick jump over to my R session here and show you how this data looks like, so that we can use our last 15 minutes to see what kind of data you typically work with. So let me here delete this. Who has already worked with R? Okay, for those who haven't worked with R, but I hope everybody has some knowledge with some programming language, be it R or Python or MATLAB or something. Everybody has some programming experience? Okay. So for those who haven't worked with R, the 1-minute summary is R is a programming language which is meant to work to be used for statistical data analysis. So it's sort of the data science language. It's a vector-oriented language. That means you typically don't work with individual values, but with whole vectors. So everything is parallelized. If you tell it add up these two variables, it automatically assumes that the two variables are whole vectors of stuff and adds them up element-wise. And you can easily work with vectors and matrices. Unfortunately, it's a fully interpreted language, so it's that slow whenever you need something complicated, but it's really convenient to use. And it has a whole load of extra packages, and what I now load here is a package for matrix mathematics, the Magritta package I explain later, and a package called Seurat. You have to pronounce it French because it's named for the French painter named Seurat, and you will see later why, which is the standard R package which is used for single-cell RNA sequencing analysis. It's developed by the research group of Raoul Satir at the Weizmann Institute in Israel, and it's sort of simply encapsulates the standard analysis that everybody does. And I will now quickly run through this standard analysis. And in the next 2 lectures, we will look at each step in detail to understand what's mathematically going on there. So the first thing I do here is that I load this count matrix, and this count matrix here is a sparse matrix. So that means it's saved in a way that, here you see the matrix and the dot means 0. So the first top left corner of this matrix is all zeros. The whole matrix has 1 row for each gene. So there's 3,200 genes and there's 2,700 cells. So each column is a cell, each row is a gene. And the numbers in it always tell us how many reads have we seen which have been assigned to this cell and this gene. And if I see a 1 here, for example, it would mean that the cell with this barcode, so with this random identifying sequence, had 1 read from this gene here. Now, the first thing we might ask is, and here you see the row names. These are all the gene names, and maybe later we will learn a bit more about how to interpret these cryptic abbreviations that biologists use. These are the column names. These are these random sequences that have been assigned to each cell, and maybe next week I'll explain to you a bit how this technically works. And now we can, for example, calculate the column sums of this matrix. And we can see here this very first gene had a total of 2,400 reads. The next cell had a total of 4,900 reads. In reality, a typical mammalian cell, and these are blood cells, so they just took the typical blood sample as a doctor always takes and sequenced these cells. And a typical white blood cell has a few 100,000 RNA molecules, but you see we only sequenced 5,000, so we lost 95% of our stuff. And we will talk a lot in the next things about how to deal with the fact that we still get reliable information despite the fact that we lost 95% of our reads. Here you can see on this histogram, where I now took a histogram of, I took these column sums, I took the logarithm to base 10 of it and then plotted a histogram. You see this thing always takes a data vector and pushes it to the next function. And you see typically most of these things have between 1,000 and 3,000 counts. We can also now pick an arbitrary cell, for example, the cell in column 1,423, so somewhere in the middle of the 2,700 cells. And what I've now done is here I said, take column 1,423 of the matrix and sort this column in descending order. So you see the gene with the most counts is B2M, which has 72 counts. Then comes the gene called MALAT1 with 65 counts. And if I do it a bit more, 200. And there's a whole lot of genes which have only 1 or 2 counts, and even many more genes which have 0 counts. We can, for example, ask how often do we see a gene that has 0 counts? You see 31,000 have no counts, 500 have 1 count, 89 have 2 counts. And those of you who have a bit of stochastics will already see where this goes. If what our first mathematical issue will be here, I told you originally there were a few 100,000 molecules, and we only see 5,000 of them. So there was Bernoulli trials happening. Each molecule had a 5% chance of being sequenced or not. So if you now ask, what's the probability of getting exactly 3 counts as opposed to 4 counts, you quickly notice this is pretty easy because there is a binomial distribution going on. And next week we will have to think a bit about what this binomial distribution that's happening here, this Bernoulli trial, the fact that we randomly pick 5% of the reads and then count, and we don't count the full complement, but a small sample of them, what this tells us about how reliable we can say that a gene with 12 counts is more strongly expressed than a gene with 10 counts.
We assume that all genes are equally likely to be counted/not?
Yes, with some caveats, which we can come later to. Actually, we don't, but we will later find an argument that this problem cancels out. So, and okay, here now we can here ask, okay, what's the most strongly we can ask what's the most strongly expressed genes averaging over all the cells. And here you see I take the row-wise averages of the matrix. And again here, on average, this gene has around 60 reads per cell. This one has 46. And most genes have less than 10 on average. We can also pick 2 cells at random. Let's take the cell in column 1352 and the cell in 762 and just plot the 2 against each other. So this here is one cell, this is the other cell. You see I took a logarithmic plot and I used this jitter function, which adds a little random value to each thing, so that all these are genes which have 2 counts in one cell and 2 counts in the other cells. And I added this little bit of random value so that they don't sit all on top of each other, and you can appreciate that really the vast majority of cells sit here. You can also see that there's a lot of genes which are high in both cells and maybe even high in other cells, while this might be a gene which is really specific to the second cell and not at all used in the first cell. So if you want to find out what is different between these cells, maybe we should look at this gene and at that gene. Now, so far I only inspected this raw count matrix as we got it out of our sequencer. Now I can run here this Sura analysis, which takes the count matrix and makes an object out of it because it's some pseudo object oriented thing. Then it normalizes the data, which means how many reads we get per cell is maybe not that relevant. If a gene has more counts in the second cell than the first cell, but in the second cell we got overall many more counts, maybe that doesn't mean anything. So the first thing we should do is that we turn the whole thing in fractions. Instead of saying this cell has got 3 reads, we should say this cell has a fraction. This gene has a fraction of 0.00032 of all the reads in this cell. That's what we mean by normalizing. Then, for reasons which we get into later, we also don't look at these numbers by themselves, but we take the logarithm. And we have to think later about why the logarithmic scale is the right scale to use here to look at these fractions. When we ask which of these genes are all the same over all the cells, and which genes differ a lot from one cell to the other, because a cell which in all genes has a gene that has more or less the same amount of fraction of reads in all the cells is probably not very helpful in understanding what's the difference between one cell and the other. This is why this function here, which again, in the next week, we will look a bit closer in what this actually means. We pick the 1,000 most variable genes in the sense of the 1,000 genes which seem to contain most information to figure out what's difference between one cell and the other. When we scale them in another way, which we'll explain later, when we run a principal component analysis on this, quick few who knows what a PCA is? Okay, for the others, don't worry. I already have, from last year, we also made a whole lecture on the mathematics behind the PCA. The core piece of which is an eigendecomposition. So who knows how an eigendecomposition works? I hope that's a few more. At least all those of you who have done a linear algebra lecture should know what it is. Otherwise, try to remember till in 2 weeks when we need that. And then, what we then do is we want to group our cells into groups of similar cells. And what we get out in the end is, let me quickly show you how this looks like, this here. Each point here in this so-called Huber plot is a cell, one of the 2,700 cells. And whenever two cells are similar, they are plotted close to each other, and when cells are not so similar, they are plotted far apart. And as you see, this allowed us to group the cells into so-called clusters, into groups of cells which are all very similar to each other. Now, the mathematicians will already notice whenever I talk about similarity, I owe you a definition of our metric. How do we define similarity? How do we quantify whether two cells are similar or not? Obviously, a cell is described by its column in the count matrix. So we have to find a metric, a distance matrix, to define distances between two such columns in the count matrix. And we will later see that simply using the Euclidean distance on the counts will not get us far. We need to do a few extra tricks to do this nicely. But in that way, we now find groups of cells which are close to each other and which are now put in one color. And now we might want to ask, what are these cells? If you're a biologist and you know that this is peripheral blood mononuclear cells, that means cells taking from blood taking out of an arm vein, and we remove all the red blood cells and all the platelets and all the strange white blood cells which have more than one cellular nucleus, and only keep the normal white blood cells with one nucleus. Then if you're a biologist, you know, well, there's there is 4 types of mononuclear cells: B cells, T cells, natural killer cells, and monocytes, I think. And which is which? And again, if you are a biologist, you might know a T cell. So for the non-biologists, that's the cells in your immune system which attack virus-infected cells. A T cell has a specific thing on its surface called a T cell receptor, which helps the T cell identify virus-infected cells. And this T cell receptor is made up out of a group of 6 proteins, one of which is called CD3 epsilon. And what we can hence do is plot this, look again at our plot, and now we color each cell up by how many counts we got in this cell for the CD3 epsilon gene. And we know these are the T cells. The same way a biologist or an immunologist knows that B cells, that are the cells which attack bacterial pathogens or which produce antibodies when you get vaccinated or things like this, that they all have a thing at the surface called CD79A. Okay, so these are the B cells, because all these cells have an appreciable count for CD79A, while all the others have 0 counts, or maybe 1 or 2, which probably were just an accident of a cell. And in that sense, we can now get these different cells. And you can already see what I have to explain to you in the next things to understand this simple analysis. What is our distance matrix? How do we find cells who are close to each other? Second, once we have our distance matrix, we get a big symmetric matrix which tells us the distance from each cell to every other cell. How do we make a 2-dimensional plot out of that? It's unlikely that this voluntarily will be a flat 2-dimensional plot, because we started off in a space where actually each dimension was one gene. You remember in the starting point I said a cell is characterized by a column of the count matrix, and the count matrix has 30,000 rows. So it's a 30,000 dimensional vector. How did we get this into a 2D places, and how many artifacts and distortions have we do we get by forcing this to be visualized in 2D. And then how do we find make a good idea of where to make the border between these colors? How do we best decide on a clustering mechanism which says which scenes should count together? To do this, we will use graph theoretical methods. And finally, we will have to talk about what to do with something like this here, where these things seem to blend into each other. Can we really say this is one cell type, this is the second, this is the third one, or is this maybe one cell type of mainly the reddish variety, maybe the bluish variety, or and in between some intermediate steps? Is this a continuum which changes from this to that, or is this three different things? And these are all questions of basically imagining that each cell is a vector in a high-dimensional vector space. We want to understand the geometry of this vector space and describe its geometry and topology. So, and this is now typically the pictures, and as you've seen, I jumped here over a lot of steps. I have here another thing where I replace each of these steps by explicit calculations, which we can explicitly see with formulas, and we will look through this in the next steps. Now, technically, as you know, in the module handbook, this was I think 4 credit point or 3 credit point things. And the math department told me that for you to earn the credits you want for this, it needs to be a 4-hour per week lecture. Quite frankly, I can only do 2 hours lecture per week, but I have scheduled also an exercise class, which is sort of semi-optional. And what I want to do there is that we simply sit down together around a big round table and do calculations ourselves in R or Python or whatsoever. Let's see how many of you turn up with this. Last year, sort of people were more interested in the lecture than the exercise classes. It also depends a bit how we do this, but I would be happy if a few people come. I'm not quite sure whether I really have material to fill a whole exercise problem class already next week, but if you're interested in redoing this kind of analysis that I've just done here in R and run it on your own computer so that you get a bit everything installed and know how to work with this data yourself. And if you have time, drop by tomorrow at 2 p.m. in our meeting room up in the 6th floor. And then we can just look a bit through this together. And then once we have a bit more material, then we can actually use this problem class to take some of these things and program them ourselves. Because it's one thing to use ready-made software and write down the mathematics behind it on a blackboard. And another thing is to actually take the mathematics from the blackboard and try to put it yourself into code. Because after all, half of you guys are scientific computing majors. And the mathematical people also should learn some programming. So let's program a bit. And so I will sort of split this, that on the Mondays I will mainly talk about math, and on the Tuesdays I will just lead you a bit in programming stuff yourself, using either R or Python or whatever is convenient for you guys. Okay, and then we will get, this is tomorrow already, and otherwise, next Monday is already Easter Monday, right? Okay, then whenever I said I will show you this next Monday, then this is of course the Monday after Easter. Yeah. Last year I did an oral exam. Who would actually need a grade? Yeah, I guess that's probably still easier to do with oral than written. Oral. Or a project. I think the module handbook is somewhat open. Let's decide all together about this in a few weeks. We can also do a project. I'm not quite sure yet. Personally, if it's not too many people, I find oral exam easy. I don't know. For the biologist, I've recently learned that you guys never do any oral exams. For whom is an oral exam something you're used to? Like who has done many oral exams already? Yeah, it's usually the mathematicians, yes, the computer science is not so much, the biologists not at all, right? Okay, we'll see how we do that. But depending on something like this. Otherwise, what else do we need? If you have any other questions, you can always ask me, send me an email and so on. If you want to look ahead, I should probably also show you this here. Here, this is the web page I've now been looking at. You see it says here 24. This will be changed in 25 once I move it over. But if you now want to already look at this, this is the 24 thing, but we'll move it to 25 because a lot of the stuff I wanted to go through and maybe improve a little bit. What else? In case we have any change of room or whatsoever, I need some way of reaching you all by email, and it seems that, has everybody gotten this when to meet mail where we made the schedule? So it seems that the mail sending feature of Heiko finally works, which means if you want to hear the things, make sure you're registered on Heiko. Which also brings me, there's always people worried about registering and deregistering for exams. Heiko has nothing to do with that. You register for an exam by simply telling me I want to make an exam, or turning up for an exam, and you deregister by simply not telling me that you want to do an exam. So no need to tell me about whether you want to come or not. Only once we make some appointment for that. Okay, then I hope to see you all in either tomorrow or otherwise in 2 weeks. Technically, I just realized next week on Tuesday, let's not do anything then. And as you might have noticed, I recorded this thing here, so maybe I upload this somewhere.